Hello everyone, welcome to our final chapter this semester. This is chapter 25. It's an integrative physiology chapter. That is to say, we're going to be using things we've learned about over the entire semester and applying it to the paradigm of exercise so that we're integrating concepts across chapters this semester. Cumulative-ish, if you will. In your academic careers, you've taken some cell biology, some basic biology, where we look at the cellular and subcellular level, some organic chemistry perhaps. Then we apply those physics, and then we apply those to the systems inside cells or whole cells, like for instance the sarcomeres in muscles or neurons and their signaling of using neurotransmitters and so forth. And then those then get applied to larger organ systems like the cardiovascular system or the neuromuscular system and so forth. And then finally we hit the highest level. We could even go beyond the highest level. We could go to multi-organisms, and that's where psychology and sociology play a role. But in any case, when we're in the clinical paradigm, we're working up here, but we're having to apply aspects of these things down here. And that's why clinical medicine and therapy is integrative. And that's why we're going to, in this chapter, we're going to help you try to make that transition to an integrative mindset, applying what you know in these earlier sections to help create therapeutic regimens and responses to problems at the organismal level. But the issue as we go from level to level to level is that each time we shift to a level, we have emergent properties. So over here on the left, this would be called a summative situation, like for instance with weights. You have weight 1, weight 2, weight 3, weight 4. Let's say you have 4 people and you're on an elevator. Their weights are going to be the sum of all the contributors. That's not really an emergent property. That can be predicted. Over here on the right, though, this is what we would call an emergent property, where it's the property of a product of interactions. Okay, the product of interactions. For instance, if I'm gaming online, I may have a core team of folks and we're doing some sort of an activity as a team. And if we're all doing our jobs and we're all relatively attentive and so forth, we have a high degree of, like for instance, success in a given task. But if somebody's busy or they're having a bad day or they're distracted for some reason, even though we have the same contributors, the product of those interactions may mean our success drops. And that's what we talk about here with emergent properties. We're taking integrated organ systems and they're interacting with each other. We're taking subsystems and they're interacting with each other. And that's why integrative physiology can be complex. We've got a few complex slides here, so make sure you take them slowly and you practice with them a little bit. This idea of integrative physiology was one of the reasons why an entire conference, 4 days, was dedicated just to this idea of the integrative physiology of exercise. Diseases like diabetes or exercise intolerance or other types of issues that we see in and around exercise are fundamentally products of interactions at these different levels. And so it's a complex topic. We're going to try to break it down for you a little bit, just show you some specific aspects. But make sure that you are taking it slow, working on material that we've seen before, because you will be responsible for some of that material that we bring up in this lecture. And here are the general topics that we're going to be talking about in this chapter. So metabolism, ventilation response, cardiovascular response. This idea of feed forward. We have talked about this a little bit, but we're going to bring it in a little bit more. Again, the idea of reinforcing the idea of positive, negative feedbacks, and now feed forward. A little bit about temperature, and then a little bit about exercise and health, including your immune system, how exercise can impact certain types of metabolic disorders, diabetes being sort of our case study. These are the pages involved in this chapter, so make sure you do a little bit of reading there as well, because you will be responsible for that material. So for our first topic, we're going to talk about metabolism. Remember chapter 4, enzymatic reactions, all right, the sum of biochemical reactions in cells and tissues. and relate it to the exercise paradigm, which we often think of as muscular, neuromuscular as well. Well, that's going to be very, very ATP intensive, right? Because ATP helps us get the job done when it comes to skeletal contraction and so forth. I would ask that you dip back into what we talked about with ATP. What were the specific roles that ATP had in skeletal and cardiac muscle? It wasn't just about contraction. We also had it for relaxation, for direct active transport of calcium out of the cytosol. We also have to maintain the resting membrane potential, not only in the muscle, but also in neurons and other cells, as a matter of fact. So make sure you remember those and recall those roles from chapter 12 and 14 and chapter 8 with neurons. Even in the most energetic of cells, though, our base supply of ATP, the ATP that is sitting around waiting to get metabolized, is very small, and it turns over constantly. You only have a couple of seconds of ATP at any given moment in any cell, much less our skeletal muscles and cardiac muscles. So a small amount, only a couple seconds worth. So we're going to have to resupply that relatively quickly. Well, one of the primary ways, especially in skeletal muscle, is substrate level phosphorylation from phosphocreatine, where we can take phosphocreatine, transfer the phosphate back onto ADP. Again, we only have a few seconds, maybe 10, 20 seconds if you're in really good shape. So it's good for short, intense exercise where you don't even need oxygen at that point. It's great for sprinting or powerlifting, that sort of a thing. If you're a swimmer, when you take off of the starting blocks, oftentimes you're underwater for a few seconds. That's a great time for these two supplies, both the ATP there and the phosphocreatine that's there. But again, a short period of time, relatively short. For our longer periods, we're going to have to dip into biomolecules, carbohydrates and or fats. Carbohydrates are great. You can metabolize them anaerobically, so you don't need oxygen. It's fast. Okay, it's certainly fast, and we'll talk about those kinetics in a little bit, but it will create lactic acid and other byproducts that have to be handled, can potentially contribute to fatigue. And again, we'll talk about those fatigue mechanisms. It is less efficient than aerobic. We're only generating about 4 molecules of ATP, a net of 2, okay, because we're burning some when we're in glycolysis. Fatty acids can't be used in this anaerobic paradigm. So a large energy supply is just not available. So this is great for max contractions for up to a few minutes, a couple of minutes, not very long. If we have to go longer than that, we have to dip into oxygen. It is the most efficient by virtue of how many ATPs per carbon we're talking about. Okay, we're talking about 34, 36, maybe 30, depending on the efficiency anyway, of ATP per glucose. All right, so it's very, very efficient relative to these other things. But it requires oxygen. And we are going to talk about differential oxygen sources in skeletal muscle a little bit later. It's great for long-term endurance because we can continually generate ATP. Not enough for maximal contraction, but certainly enough for sustained above basal level metabolism. And just a review slide for you here about phosphocreatine. Remember that creatine kinase controls both directions. Need for speed, right? Need for speed. Here are those 3 big roles in skeletal muscle to remind you what ATP is involved directly. These are directly involved with neuromuscular activity of exercise. Never mind other biochemical and metabolic needs. And another review slide on the major causes of muscle fatigue during exercise. If we're talking about long-term exercise where we're doing aerobic, one of the issues, especially if it's fairly intense, above let's say 45 or 50%. So we're still submaximal, but we are cranking pretty hard. The issue here might be glycogen, because in submaximal, you may be beyond what you can generate aerobically. And so you're dipping into anaerobic, which means you're dipping into glucose, and glucose is stored in glycogen. If we're talking about maximal exercise, inorganic phosphate release seems to contribute to immediate, when we're talking about true maximal, true anaerobic level contraction. Calcium release may also play a role, but probably the number one thing is the release of inorganic phosphate from myosin, and that's why we went through that power stroke figure in the last unit to have you guys understand that concept of why we might have plenty of ATP, we might have plenty of glucose, but why does that muscle fatigue? It's not because of lack of oxygen, all right? It's often due to the other biochemical mediators, including inorganic phosphate released from ADP. Doesn't come off, slows the kinetics of myosin, which means less power generated. Certainly calcium can also be a role there as well. Maximal exercise, this is maximal extended exercise. We can have issues with potassium, altering membrane potential, changes in sodium potassium ATPase activity. All right, so these again are a little bit of review, just bringing this back from previous lectures. And again, this is just another figure for you. All right, central fatigue, that's where lactic acid often does its thing. You know why? Because the body can use lactic acid to generate additional glucose, and I'll show you that in just a moment. All right, so remember these sort of major areas of fatigue. All right, central fatigue primarily driven by nociceptors and lactic acid buildup. These other things down below, here's our calcium, all right? Calcium release can be an issue at what we call peripheral fatigue, but also inorganic phosphate buildup, all right? So keep those in mind when we think about how we're going to address those if someone has exercise intolerance and we know that it's a function of the muscle. Quick word of warning about this slide, and that's why I've got the Achtung here, because this slide is complicated. There's a lot going on here. We're going to be integrating concepts that we've talked about all semester. So we're going to take it slow, try to work through the different landmarks. I am going to work through 3 first rather than 2, but in any case, I recommend that you print this slide out from the PowerPoint and annotate it or draw a stick figure to kind of keep, because there's, again, lots of simple things, but when you put it all together, it makes it feel and is complicated. Emergent properties, that sort of a thing. Well, we've got muscle contraction going on. That's what we're going on during exercise. This is muscle. This is skeletal muscle. And so during exercise, we're cycling through ATP and ADP, PI in order to do some of those things that we talked about, in terms of keeping the cellular ATP pools at a certain level, creatine kinase and so forth over here. Well, how do we sustain ATP or regenerate it? Through things like glucose. And glucose can be absorbed from the digestive tract into the bloodstream and then into the muscle, or we can get glucose from liver. The liver will take up glucose during times of rest and make glycogen. Same thing is true over here, that if the skeletal muscle brings in glucose and doesn't need it, it will store it to a given degree in glycogen. But as soon as exercise kicks in, we're bringing in glucose and we're going to metabolize it. Well, if we have glucose, and there are other energy sources, we'll get to those in a moment, glucose has to go through glycolysis, whether or not we're going to be oxidative or not. All glucose, unless it's being used for some other purpose, has to go through glycolysis. Now, but we also have different kinds of muscle, right? We have slow twitch and fast twitch and those sorts of things. So if it's slow twitch and there's enough oxygen, we can undergo oxidative phosphorylation. Or if it's that medium twitch, if you will, remember the ones in orange here are what we call facultative. That means that if there's enough oxygen, they'll undergo oxidative phosphorylation. But if there's not, they'll stay with glycolysis and make lactic acid. If it's a truly energetic contraction, we're going to be full-on anaerobic, making lactic acid. Now, the fun part about lactic acid is that it can be recycled. And this is sort of the new twist here. That lactate will contribute to central fatigue. Remember, we talked about it. It'll activate nociceptors and will contribute to the brain not losing its resolve to undergo exercise, right? But we also can recycle lactate back into glucose. We can manufacture glucose in the liver from lactic acid. We can also manufacture glucose from amino acids, and this process is collectively known as gluconeogenesis. Genesis meaning the origins thereof, new glucose. New glucose creation. So either synthesizing from amino acids, all right, like glutamine, all right, or glycine. We can also manufacture glucose from lactate. So lactate is an important signal, but it's also the source of new glucose. We can also use glucose in oxidative phosphorylation. We all know that, but there's another entry point. Fatty acids, and fatty acids can come from either the fat tissue. We can release fatty acids from lipids stored in adipose. We can also absorb fatty acids from the intestine in some cases. In both cases, though, fatty acids are turned into acetyl-CoA with that beta oxidation. Remember beta oxidation? We make acetyl-CoA, and then we can make ATP from that. But fatty acids, or if we're undergoing oxidative phosphorylation of glucose, require proper gas exchange. We have to have enough oxygen. Now, oxygen does come from the lungs, and we cycle the gases that way. But oxygen can also be stored in the muscle, and we'll come to that a little bit later as well. Again, a lot going on here. If I were going to give you a figure like this on the exam, I wouldn't give you all the information in this figure, but I would give you the roadmap and ask you to explain what's going on in your own terms. So practice using the terms of physiology, the terms that we've used, and try to demonstrate that your understanding of what's going on in these different places. Which energy sources go through glucose? Which sources are solely aerobic and which can switch between aerobic and anaerobic? Again, a semester's worth of material here integrated into one figure. So there you go. In your book, you have this figure. This is figure 25.2, and it's showing you the relative rapidity with how we can create ATP. If we have to use the non-ATP sources to create ATP, we've got phosphocreatine, we've got anaerobic metabolism, and we've got aerobic. In the salmon color, that is how much ATP, moles per minute, we can create. And you can see that phosphocreatine can create a boatload of ATP in a relatively short time, but for only a short time. So we can make a lot of ATP this way, but only for a very short time. All right, if we can make 4 moles of ATP in a minute, but we only have 10 seconds, so that's 1/6 of a minute. Okay, so what's 1/6 of 4? It's going to be somewhere under 1. So, all right, so keep that in mind. Yes, we can make it really, really quickly, just so we can't do it for very long. Anaerobic, not quite as much ATP in unit time as, say, the phosphocreatine, but it lasts a little longer. It says 1 minute here. I would argue that if you're in really good shape, you can push that longer, closer to 2 minutes. Okay. Again, not quite as fast as phosphocreatine, but we can sustain it for an extended period, relatively speaking, extended period of time. And then over here on the right, you can see that we can't produce a lot of ATP. We can produce it, just not very quickly. That's the point here. This is ATP over time, all right, 80 moles per minute. So it feels like it's not producing that much, but it actually can produce a whole bunch. You know why? Because it can go on for hours and hours and hours. So while we can't create it as fast as these other two, we can create it for orders of magnitude longer periods of time. 1 minute, I mean, if this was just 1 hour here, that would be 60-fold of this. But we're talking about multiple hours, sometimes in the excess of 6 or more hours. So while it's not as fast, it is sustainable. And so here's sort of the relative rate. If we set phosphocreatine at 100%, You can see that glycogen and anaerobic metabolism is somewhere between 1/2 to 1/4. All right. Fatty acids only about 10%. All right. So relative rate of ATP synthesis. And here showing you the sort of time, again, showing this in terms of time. So ATP, we only have a few seconds. So percent maximum drops very, very quickly if all we have is ATP. If we have phosphocreatine, we can supplement, all right, up to perhaps a minute or two. Lactic acid, here's lactic acid in here. I think that's kind of interesting. That's because we can convert lactic acid back into glucose. And then finally, aerobic. And this is why it takes a few minutes. If you want to work in aerobic, you have to kind of warm up for 5 or 6 minutes. That's because what we're doing is we're ramping up the aerobic system that way. And then you have overall ability, strength of muscle contraction over time. You can see it drops off as we get away from the anaerobic situations and into the aerobic. Again, endogenous ATP, phosphocreatine, lactic acid, and then lastly, the oxidative phosphorylation. Where can we take all of this information that we have learned this semester and apply it in a relatively simple way for clients, loved ones, folks we care about? Presume for a moment that our person in question that we're helping is somebody who needs to lose weight. Well, we're going to be using energy sources and burning calories, and that's important. But how we burn the calories becomes really, really important. And so here we have 25-3. It's really, really interesting. Remember that we have oxidative fibers, we've got oxidative glycolytic fibers, those medium twitch, and we've got the true fast twitch, the fast glycolytics. Again, we recruit those depending on percent effort. If we are at rest, we can use whatever energy source we want, but as we increase our intensity, we're going to be recruiting more and more fast twitch type things that are going to be using carbohydrate. So when we're in the relatively low intensities, up to about 45 to 60%, somewhere in here, what happens is most of our energies are going to be coming from acetyl-CoA, from beta oxidation. So most of our energy is going to be coming that way, with a little bit of carbohydrate mixed in there as well. All right, so that's carbohydrates. That's glycolysis and/or creating oxidative processes. We're not doing a lot of anaerobic here, but as we get closer and closer to around this 45 or so, we're going to start recruiting some of those anaerobic fibers. All right. And that's what's happening. We're going to have a break point here where more and more we're going to be going into glycolysis and out of beta oxidation. So if you have somebody that wants to lose weight, what we're going to need to do is tailor their exercise so they're not in this paradigm out here. They're in this paradigm where we're burning more fat. Because fundamentally, obesity is about too much body fat. All right. You can have very, very fit muscles and still have a high BMI. That's been known for a long time. But how do we get those calories that are stored in fat out? Is that we utilize fat energy reserves rather than carbohydrate energy reserves. And that's sort of what's shown here. This is sort of that same figure, only is showing you power output in terms of watts, if we were measuring them on a bike or a treadmill or something, but it's basically the same graph. As we get to about the 50 to 60% mark in terms of max power, what happens is as we go there, we actually turn off oxidative systems. We almost go exclusively into carbohydrates. And since we only have about a pound of glycogen in our bodies anyway, that's not going to be very productive for weight loss. Where we want to be is somewhere out here in the 40% range, in the 40% range for much longer periods of time. All right, you go longer at lower intensity is better for weight loss than faster at high intensity. And maybe many of you guys understand that principle, but now you have the cellular biology to explain this. And that's the difference between you and everybody else out there. You have the physiology and the cell biology to understand why this is the way that it is. Your body needs ATP, and so it shunts into an anaerobic paradigm. All right, shuts off the oxidative paradigm. Where we get our maximum oxidative is somewhere out here in the zero, in the 35 to 50% range. It's not just about effort, it's also about hormones. So hormones are involved. We know about insulin, we'll talk about glucagon and some other hormones here as well. There are hormones that are pro-energetic, and one that we're going to talk about is called cortisol. All right, it is actually the right amount of cortisol during the right exercise paradigm, and you can see these folks are exercising in a low intensity way with longer duration. So lower intensity, longer duration, because their goal is to probably be either and or weight control and or glycemic control. So we want to be converting triglycerides into fatty acids for beta oxidation. So cortisol is a big one of those, all right? It's involved in that. It's a stress response hormone, but catecholamines, the sympathetic system. Remember chapter 11, sympathetic systems, catecholamines, epinephrine and norepinephrine, growth hormone. We often refer to that as human growth hormone. You might hear the phrase HGH, excuse me. Those are all known to trigger lipolysis, if you will. That is the conversion of triglycerides and fats into their original products, glycerol and fatty acids. When we convert glycogen, we can sustain glucose levels in the body. So if you exercise with a modest diet, what's going to happen is that's going to promote glycogenolysis, that is the breakdown of glycogen in order to keep glucose levels in a homeostatic window. Because your heart and your kidneys and your brain all need glucose all the time. Your muscles and other organs can get away without glucose, but we have to sustain those glucose levels for those organs specifically. So we can convert triglycerides when we're going in moderate exercise. These hormones also will sustain glucose levels if our glucose levels are dropping. So glucagon's a big one of those. But again, catecholamines can do this. Cortisol can do this as well. These guys are minor players. This guy's the major player. Again, back to chapter 7, our endocrine system. Now, during exercise, we typically will suppress insulin, which on the surface seems problematic. We've got this glucose coming into our bloodstreams. Well, how are we going to get it into the muscles? And we always know that glucose stimulates muscle absorption, or insulin stimulates muscle absorption of glucose. Well, it turns out that we can reduce glucose uptake in some cells because there's no insulin there. So but what it's doing is it's preventing unnecessary glucose uptake. We're exercising right now, so we don't need certain types of organs functioning. For instance, digestive organs don't need to function all that much during exercise. All right? So some cells are going to have lower uptake, but muscles have a separate signal for uptaking glucose that doesn't require insulin. Okay? We know insulin's role in glucose uptake, but during exercise, we know that insulin levels drop. Certainly some cells are not going to take up as much glucose. Again, unnecessary uptake. But we still need glucose in those muscles, and so the glucose has a secondary signaling system for taking up glucose in the absence of insulin. That's going to dovetail back in a little bit later. So we've alluded to some hormone stuff like in the previous slide and some metabolic stuff in the slide before that. But what are some limiting factors, potential limiting factors of exercise? Certainly people that are in good shape can exercise a lot more than folks who can't, people who have their cells are acclimated, if you will. But it also can come back to that original metabolism. That is to say, we are consuming oxygen during sustained exercise activity, and we can measure that indirectly through oxygen consumption. Without oxygen, we can't undergo Krebs cycle and oxidative phosphorylation. We would be forced into lactic acid. But oxygen consumption also continues even after exercise is done. So like these two folks right here who are resting after exercise, what's happening is we're using oxygen in order to restore our creatine phosphate, to restore oxygen levels in a secondary oxygen delivery system to the muscles. And that secondary system, sort of like glycogen is to glucose, myoglobin is to oxygen. Myoglobin is unlike hemoglobin in that it's one subunit, one heme, but it is a molecule inside muscle fibers that can store oxygen. Not unlike creatine kinase and creatine, or like I said, glycogen and glucose to a certain degree. Myoglobin will absorb oxygen during periods of rest and release oxygen during periods of exercise. And just for fun, this is not in this chapter, here's our hemoglobin saturation curve. Remember that, our affinity curve? Well, let's apply myoglobin to it, and you can see that myoglobin is way left shifted relative to hemoglobin. When it's left shifted, it means it has higher affinity for oxygen, okay? But myoglobin affinities can shift just like hemoglobin does, but I wanted to show you this in the resting, in the regular state. This would facilitate delivery of oxygen to the muscles, okay? It helps keep oxygen concentrations relatively low compared to the bloodstream, which means that hemoglobin will be releasing oxygen to muscles. And so here's an application of that hemoglobin saturation curve for myoglobin. But we are still working through this idea of post-exercise. We're restoring ATP, we're restoring myoglobin, oxygen, and so forth. That ability to obtain and use oxygen, whether it's inside the muscle, okay, the ability to regenerate, or the ability to deliver. Folks that are not as well acclimated to exercise will have fewer mitochondria than folks that are in good shape. Fewer mitochondria means less ability to make ATP. Maybe you don't have enough blood supply in terms of the vasculature. We know for a fact that well-exercised muscle has denser capillary beds than unexercised muscle. So maybe some of the limiting factors of exercise, we can't get oxygen there because we don't have enough blood vessels. It's not limiting. The ability of muscle fibers to use oxygen is not limiting for maximal strength. So somebody who is not in great shape may still be able to generate a huge amount of force for a very, very short time. They still may be plenty strong. They just can't sustain it. Okay? That's the idea here. That's because maximum contraction is anaerobic. Okay? It's anaerobic. We don't need mitochondria for anaerobic. We just need enzymes in the cytosol for anaerobic. All right? Don't need oxygen. Just need glucose and ATP. So it's not limiting for maximum exercise capacity, but it is limiting for sustained exercise capacity. So it's influencing sub-maximal. Our last slide for this part of the video is showing the relative lag between oxygen consumption and exercise. What can happen is that as we start exercise, oftentimes we are exceeding the ability to the stores of oxygen in the myoglobin as well as the delivery capacity of the vasculature. That's because cardiac output hasn't bumped up, and we'll talk about that next time. So there's a little bit of lag there post onset of exercise, but there's also a lag post-exercise. We just talked about that in the previous slide. So what does this graph mean? It's that we're often going into deficit here and the body responds, and then we lag a little bit over here as we regenerate stores of ATP, for instance, creatine kinase, or oxygen and myoglobin. And that's why you continue to have rapid respirations and heart rate for minutes or long, for longer than the exercise ensues. This concludes part 1. We will wrap up the rest of chapter 25 in part 2. Thank you for your time, and as always, if you have questions, please just let me know, and I'll be glad to get back to you as soon as possible. Thanks. Talk to you soon.